Hello, AP Bio. In this video, I'm going to talk about the patterns of inheritance, but all of classes are now in different places. So some of you didn't get the very end of Mendel meeting Punnett. So if you already got this part in class, you can zip right ahead. If not, then you should watch this part to finish the bottom of page 15. So I'm on page 15 in your notes, and I'm here in the class presentation. And just to refresh your memory, Mendel and Punnett got together. Punnett was this mathematician guy, and he said, oh, inheritance is a game of chance. If everything's diploid, then it's really just the same thing as flipping a coin. You have 50% chance of getting maternal and 50% chance of getting paternal. So that's why it's this flipping a coin for every single locus. So for every single gene, it's a 50-50 chance. And Mendel took Punnett's mathematical ideas and he applied them to his thinking around crossing individuals to try to determine the patterns of inheritance. And he had all these different types of crosses. We talked about one trait crosses in the class. That's just two parents and you track one character. That's why it's called a one trait cross. A monohybrid cross, this is written a little bit confusing here. Technically, in a monohybrid cross, what you have is you have two true breeding parents in the P generation, and then you get all hybrids in the F1. And a monohybrid cross in Mendel language is you take that F1 generation and you cross it. But we can think of it just as, you can forget about how you develop the monohybrid cross and think about what the monohybrid cross actually produces. It's what comes out of this cross right here, which is what's in the F2 generation. When you cross two hybrids, that's what a monohybrid cross is. See, this one is hybrid, heterozygous, we can call it also. And this one is hybrid, also heterozygous. You're going to get in the F2 generation, which you can see right here, a 3-to-1 ratio. So you'll have, really, if you look at what the cross is, you have homozygous dominant here in this individual. And then in these two, they're both heterozygous, but they present with the dominant phenotype in when we talk about complete dominance, in a situation where there's complete dominance. And then here in this 25% of the time, we get the recessive phenotype that pokes back out in that F2 generation. So a monohybrid cross you can think of as just a cross between two heterozygous individuals, and whatever that outcome is, you're always going to have a 3-to-1 ratio in that generation, the offspring of these two parents right here. Now, third type of cross is a test cross. And in a test cross, we're trying to figure out, we're testing, that's why it's called a test cross. We're trying to figure out what the genotype of the individual being tested is. Well, you'd never do a test cross if an organism has the recessive phenotype, because that can only be little a, right? Because that's the recessive phenotype. But you would do a test cross if you have an individual who is expressing the dominant phenotype, because the dominant phenotype is actually a mix of two different genotypes. It could either be homozygous dominant or it could be heterozygous. And because of that, you can do a test cross. And all that you do with the test cross is you cross whatever you're testing, this individual that you're testing, and you cross it with a homozygous recessive individual. And that should reveal whether or not you have your test individual is heterozygous or if it's homozygous dominant. And that's another type of cross. The last type of cross that we're going to talk about, two types of crosses, I guess, are a two-trait cross and a dihybrid cross. A two-trait cross is the more general term, and it's the same thing as a one-trait cross, except now we've got two characters. So it's a cross between two parents again, but this time we're tracking two characters. So in the case of this diagram right here, we would be tracking the color of the pod and then also the color of the seed. So seed color and pod color, you could track those two traits. Now, a dihybrid cross is a type of two-trait cross. And again, it's Mendelian language, and specifically what a dihybrid cross is it's a cross between two individuals that are both heterozygous for two different characters. But in both of those characters, they're heterozygous for the trait. So in this particular case, this is showing you a dihybrid cross. Let's see why. Well, the pod color is capital GG hybrid, But then also the seed color is also hybrid. You can see it's hybrid for both. So in a dihybrid cross, this would be the one at the first individual, the male on the top, big G, big Y, little y. They're both hybrid, both heterozygous for both loci. And also the second individual in the cross too is both heterozygous for both characters. So you have to have, if you think about it, if you're crossing two individuals for two traits, there's 4 different loci that you're looking at, 2 from one individual and 2 from the other individual. And a dihybrid cross, all of them have to be hybrid. All of them have to be heterozygous. Now, when you have that condition, if you cross 2 dihybrid individuals, then you get in your offspring, you get a 9:3 to 3:1 phenotypic ratio, which means if you looked at the individuals from that cross, these individuals here inside this Punnett square, you would see that in a ratio of 9, you'd have dominant phenotype, green pods and yellow seeds. You can see it. 1, 2, 3, 4, 5, not this one, 6, 7, 8, not these, 9. And then in the ratio of 3, you would have the dominant phenotype for the first character, and in the second character you would have recessive. So dominant for the 9, dominant recessive phenotype for the second. So let's look and see what that means. Well, that would be a green pod, that's dominant, but a green seed, green. So let's count them. 1, 2, 3, and then flip-flop that for the next 3. So the next 3 would be a recessive phenotype for the first locus, and then for the second locus it would be dominant. So that would mean in the case of this diagram right here, we would be talking about a yellow a yellow pod with a yellow. So this would be that 3, and we have 3 of them. 1, 2, 3 for yellow, because remember, it's weird, it's the yellow seed is dominant. And then finally, in one, only 1/16 of those would be recessive with the yellow pod and the green. We have that 9:3 to 3:1 ratio in a dihybrid cross. Okay, let's go on now to patterns of inheritance. Those are all the different types of crosses. Now we're going to talk about what results from those crosses. That's what we call patterns of inheritance. What does that look like? And the one that we've been talking about so far is just complete dominance, which is part of Mendel's model, the law of dominance. And so that's why we call complete dominance. This is also called Mendelian inheritance. And in Mendelian inheritance, or in complete dominance, we have the dominant allele determines the phenotype. The recessive allele is going to be masked by that dominant allele, and you end up with 2 distinct phenotypes. You only have the dominant phenotype. That's the case either if it is homozygous dominant or if it's heterozygous. That's the same phenotype. Or you have the recessive phenotypes. You have 2 distinct phenotypes. And in this case, you have, we can have an example here I've shown is a hairline. So we use a capital W for or a capital letter for the dominant allele. In this case, it's capital W for widow's peak. And we use lowercase w or a lowercase letter, a lowercase symbol for the recessive allele. In this case, it's straight hairline. So this is, again, Mendelian inheritance is what we all know about, or many of us know about. And it's also called complete dominance. And he called that the law of dominance. It's part of his model. But the thing is it's actually not quite that simple. And there's all sorts of discoveries that have been made since Mendel's time that we call non-Mendelian inheritance, because not all characters are going to have that same pattern of inheritance. There's lots of different mechanisms. And so let's talk about those now. These are not any less important, and it's not that Mendel's studies are not important. It's just that there's more to the story. He only told a portion, one chapter of the story. So let's talk about the other chapters, the other patterns of inheritance. The first 2 I want to talk about, I consider degrees of dominance. In complete dominance, we talk about how the dominant allele totally masks the recessive allele, but that's not always the case. Sometimes the dominant allele, there actually isn't one, and both alleles kind of poke through and can be shown in a phenotype. In a pattern of inheritance called incomplete dominance, you have the heterozygous genotype is expressed as an intermediate. So therefore we have three different phenotypes because you have the dominant phenotype, you have the recessive phenotype, and then you have the heterozygous phenotype that is like in between. And in the case of incomplete dominance, the heterozygotes appear somewhat blended because neither phenotype is going to be expressed. Classic examples are Andalusian chickens, which are these chickens right here. It's such a weird, I don't know why that's an example, but that's an example, where Andalusian chickens can either the feathers can either have the white allele, they can have the black allele, or there's this heterozygous condition where they're blue, and you can see what a blue chicken looks like right here. We also have 4 o'clock flowers or Mexican sunflowers is another classic example. Now, this example I'm showing you here is straight out of your textbook. I really don't like it because I don't like the symboling system that's being used. The symbol system, you should stick with the Mendel system and take it one step further. So anytime we're not talking about complete dominance, you use superscripts in any of the degrees of dominance to show that it's not Mendelian inheritance. And so in the case of incomplete dominance, because neither of them show through, I think of them as both being recessive. So you should use a lowercase superscript. And in the case of, let's do 4 o'clock flowers over here, this 4 o'clock flower white would be F superscript little w, F superscript little w. And then over here, it would be F superscript little r in both of these positions here. And then all of the heterozygotes would be a blend, in this case, pink. And so you would write it F superscript little lowercase r, F superscript lowercase w. Keeping in mind that I typically recommend writing them in alphabetical order, just helps you stay organized. When the problems get more complicated, you want to stay in alphabetical order. Okay, the next pattern of inheritance is codominance. Codominance is similar to incomplete dominance, where no one can decide who's going to talk, as they do in the instance of Mendelian inheritance. But in this case, it's more like co-chairs or co-presidents. Everybody's strong and everybody has a voice. And you can think of that with terminology, co, right? They're both actually going to be expressed, and that's actually what happens. If you're evaluating a population and analyzing a population and you see 3 phenotypes, you know it's not Mendelian inheritance, because in a Mendelian inheritance, you can only have 2 phenotypes. And so we have, in codominance, the heterozygous genotype is expressed as both phenotypes. And so therefore, there's actually 3. Again, there's the dominant phenotype, there's the recessive phenotype, and then there's the both phenotype, the heterozygous phenotype. So both alleles are going to express co. You can think of it as, like I said, co-presidents or co-vice chairs or whatever. In any case, the classic examples of this are cattle coat color and the AB blood type. Here we have a photograph of cattle coat color. So cattle coat, the allele that codes for coat color can either be the white allele, or it can be the red allele, or in the heterozygous condition, both red and white are both expressed, and we call it roan. R-O-A-N, roan. So this is what a roan cow looks like, and you can see that both phenotypes are expressed. In the case of AB blood type, here's a red blood cell. In type A blood, that's a surface protein that's expressed, the immunoglobulin protein is type A. In type B blood, you have the B immunoglobulin expressed, type B. But in type AB blood, they're just both expressed. So you have a codominance, a case where both of those proteins are going to be expressed in the surface of red blood cells. Now you'll see here a sneak peek at looking at one of the next patterns of inheritance. Here you have a blood type O blood that has neither expressed. So here you can already see that there's actually more complexity. We'll talk about that in the next type of non-Mendelian inheritance. Okay, that's it for codominance. They're both expressed. Now let's talk about multiple alleles. In multiple alleles, what we have is, we'll stay on this slide right here to talk about it. Most loci, the overwhelming majority of loci actually have multiple alleles, meaning that there's not just 2 choices like Mendel had in his research. There's many, many more. And in the case of blood, we have the A allele, the B allele, and then the O allele. And when we talk about all the different alleles well, first of all, multiple alleles just means that there's more than two alleles for a given gene. And like I said, most characters are going to have multiple alleles. But when we talk about all the collection of the different alleles, we call it an allelic series, which just is a collection of all the alleles for a given locus.